So hi everyone, today I'll be presenting our results that were published at the end of last year on the genetic basis of adaptation and speciation. I acknowledge the traditional owners and their custodianship of the lands in which we meet today and pay my respects to their ancestors and their descendants. So in this talk, I'm going to give you some background about adaptation and speciation, which leads into our published results, which I then want to follow up with our current work to give you an idea of where we're heading with some of our research in the centre. So natural selection is a major driver for the diversity we see in nature, from the diversity of beak morphology in Darwin's finches to the diversity and specificity of plant-pollinator relationships. Natural selection not only drives the evolution of traits that enhance fitness to the surrounding environment, but can also directly lead to the origin of new species. So understanding these two processes and their connections is key to conserving, utilising, and knowing how diversity arises and persists across the world. So as you know, adaptation is evolution that enhances survival and reproduction in that environment. For example, populations have evolved differences in coat coloration to camouflage from predators in different environments. So this has occurred in hares, lizards, mice, and many other species. These adaptations have improved their fitness in these environments. On the other hand, speciation is the evolution of distinct species, and it occurs as reproductive barriers evolve between populations. An example of a reproductive barrier is hybrid sterility. Mules are hybrids between horses and donkeys, but they're a dead end, evolutionarily speaking, as they are sterile. As a consequence, genes from donkey population cannot be transferred to a horse population and vice versa. So there are many reproductive barriers, and they can be grouped into extrinsic and intrinsic barriers. Extrinsic reproductive isolation is driven by divergent natural selection as populations adapt to different environments. So this form of selection pulls apart the two populations. In one great example of extrinsic reproductive isolation, Linden and colleagues discovered that deer mice that don't camouflage with their surrounding environment are more likely to be spotted and eaten by a predator. In this case, eaten by this cute owl. So if the mouse dies before it can reproduce, then this is a barrier to reproduction created from its coat color. This and many other examples have demonstrated that populations adapted to their local conditions create various forms of extrinsic reproductive isolation, therefore driving speciation. So in general, ecology plays a fundamental role in creation and maintenance of biodiversity. In contrast to the ecology of organisms, some processes like genetic conflict or sexual conflict can evolve independently from the environment that a population occupies. As these forces drive the evolution of traits within populations, they incidentally create reproductive isolation between other populations. So we refer to these barriers as intrinsic reproductive barriers, and the most common ones are hybrid sterility, like we observed in the mule, and inviability, where hybrids die before sexual maturity. So they arise from the accumulation of mutations that work within a population, but not between populations. So let me explain this in visual form, using the Bateson-Dubansky-Miller model for hybrid incompatibility, where two populations split from an ancestor and new mutations arise and spread in the separated populations. But there is a catch. When these new mutations are brought together, they are incompatible and hybrid failure occurs. So these failed interactions in alternative genetic backgrounds are known as genetic incompatibilities. So the evolution of these new mutations can be driven by different evolutionary forces like drift, sexual selection, and natural selection. And although there is evidence for some of these forces in driving the evolution of genetic incompatibilities, it remains unclear if some forms of natural selection can drive this process. In particular, we still don't know if local adaptation, as Darwin envisioned, truly can drive the evolution of not only extrinsic, but intrinsic reproductive barriers. In other words, are alleles responsible for local adaptation also contributing to hybrid failure? There are only very few examples that link local adaptation with intrinsic reproductive isolation, and therefore the details of how selection creates intrinsic reproductive isolation remains elusive. So with this in mind, can local adaptation drive the evolution of intrinsic reproductive isolation? Such an answer is key to our understanding of how biodiversity arises and persists. So to address this question, I have used the Senecio lautus species complex, which is a native Australian wildflower where divergent traits have evolved in contrasting environments. In the dune environment that is sheltered and has low salinity, an erect growth habit with few branches has evolved. And in the headland environment that is windy and has high salinity, a prostrate growth habit with many branches has evolved. These traits are maintained when they're grown in the glasshouse, indicating they have a genetic basis. So these dune and headland populations often occur adjacent to each other across the eastern and southern coastlines of Australia. So this phylogenetic and other analyses by Manny James shows that dune and headland adjacent pairs are often sister taxa, so they share a common ancestor, indicating that these contrasting phenotypes between dune and headland plants have evolved independently multiple times. In other words, we can study adaptation and speciation in natural replicas of the process, which is incredibly powerful. More research done in our lab has shown that genes with predicted function in certain hormonal pathways were repeatedly differentiated between adjacent dune and headland populations. However, these genes need not be the same in each population pair. For instance, a major signal of differentiation in these dune headland pairs came from different genes but with predicted functions in the auxin pathway. So it seems that there are many solutions to the same problem, but they can be different genes but connected by a common functional pathway. So why are these auxin genes repeatedly differentiated? What is auxin? As many of you know, and much better than I do, auxin is a hormone that is transported from cell to cell for cell growth and differentiation. So one major role is to alter the direction of growth of a plant. For example, redistribution of auxin after a stimulus such as light or gravity induces bending of the stem. Could the auxin pathway contribute to the genetic basis of adaptation in Senecio lautus? In other words, we wanted to know whether genes within the auxin pathway were repeatedly targeted by natural selection to enhance fitness to these contrasting environments. And we can test divergence in the auxin pathway by examining divergence on phenotypes dependent on auxin, such as shoot gravitropism, which is the ability of a plant shoot to bend upwards in response to the downward pull of gravity. As mentioned before, auxin redistribution to one side of the shoot is required for a gravitropic response. So let's see this in greater detail. If a seedling were rotated by 90 degrees, auxin would be transported to the lower side of the shoot, which would create a high concentration of auxin. This localization of auxin to one side of the shoot will induce elongation of cells and therefore causing the shoot to bend upwards. So this is known as a gravitropic response, and this is an easy phenotype to measure to look at divergence in the auxin pathway. So this leads me to 4 research questions that I'll delve into in the next part of the talk. So I'll start with my first research question. Has gravitropism diverged between adjacent dune and headland populations? This will give us an indication of whether natural selection has repeatedly targeted the auxin pathway in independent populations. So to investigate this, we use 16 natural populations of Senecio lautus that occur across the eastern and southern coastline of Australia. So this includes 7 dune and headland adjacent population pairs and 2 alpine populations, where one has a sheltered environment like the dune and has an erect growth habit, and the other has an exposed environment like the headland and is prostrate. So I measured gravitropism in these 16 populations by rotating a 7-day-old seedling in an agar plate by 90 degrees and measuring the response angle after 24 hours. So the bigger the response, the bigger the angle. And here are the results for the gravitropism in these 16 natural populations. So the dune population is on the left of each pair and the headland on the right. The dune population has a higher gravitropic angle than its adjacent headland pair in most population pairs, showing that gravitropism has diverged between many adjacent dune and headland populations. So now I have added height measured in the glasshouse to the figure. And as you can see, the dune population is taller than its headland pair in most populations, and this often correlates with what is observed with gravitropism. For example, these 5 pairs within the green box, which includes the alpine populations, all have the dune population being taller and more gravitropic than their adjacent headland population. So the other pairs either do not show major differences in height and gravitropism, the blue box, or show dramatic differences in height, but not in gravitropism, the purple box. So these results show that height and gravitropism have evolved in parallel across many, but not all coastal and alpine populations in this system, suggesting that alternate pathways might be utilized in these other populations. So now that we have established that gravitropism has evolved repeatedly and independently in many natural populations, I investigated whether gravitropism contributes to local adaptation in an advanced recombinant population. So we created a recombinant population to reduce genotypic and phenotypic associations so we can find the genetic basis of key traits. So we used 45 parental dune and headland families from Lennox Head and completed random mating with equal contribution for 8 generations to create an F8 recombinant population. So basically this population is perfect for associating genotype, phenotype, and fitness, because many of the associations expected to occur in nature would have been disassociated with random mating. So this F8 recombinant population was planted into the parental dune and headland environments at Lennox Head in New South Wales. So the seeds were glued to toothpicks and placed just under the soil surface in a randomized block design. So we performed family-based selection for 3 generations, so in the F8, F9, and F10 generations, and produced an F11 generation that we use in the following experiments. So this time gravitropism was measured in pods so that we could measure other traits in these plants like height at maturity, and also so we could collect healthy leaf tissue for DNA extraction. So we rotated a 13-day-old seedling by 90 degrees by putting their pot on the side and measured gravitropism 12 hours later at the end of the dark cycle. So this is what gravitropic distribution looks like with agravitropic individuals that don't respond well to gravity stimuli on the left and gravitropic individuals that respond well on the right. And these are the results. So we have maternal fitness is on the x-axis and offspring gravitropism is on the y-axis. So we found that fitness of the maternal parent in the dune environment predicts offspring gravitropism. So we found no association with paternal fitness. So it is the maternal genotype that is driving the association with gravitropism. So high fitness of the mother leads to offspring with a higher gravitropic angle after selection in the dune environment. In the headland environment, we found no correlation between fitness and gravitropism. Instead, in the headland survivors, we found a correlation between individuals that die early in development in controlled conditions and those that have a small gravitropic angle. We therefore believe that there could be intrinsic reproductive isolation occurring with agravitropic alleles being incompatible with a hybrid genomic background. So the key finding from this data is that dune survivors produced offspring that were more gravitropic. And overall, this indicates that gravitropism, or a tightly correlated trait, contributed to local adaptation in this environment. We then wanted to know whether gravitropism was the target of selection, or a tightly linked trait like maybe height was driving its evolution. So as I described before, there is a strong correlation between height and gravitropism across many natural populations of Senecio lautus, where dune populations are tall and largely gravitropic, and headlands are short and largely agravitropic. So this is perhaps not surprising to some extent. So there is likely an adaptive advantage of being short in the windy headland environment, where branches break easily. So we first asked whether these 2 traits are tightly linked in the genome. In other words, it is possible that variation in the same genes contributed to variation in both traits. It could also be separate genes, but right next to each other in the genome. So these scenarios would create a genetic correlation between height and gravitropism. Alternatively, it could be that natural selection just targets these 2 traits independently as they provide a fitness advantage, and they are not connected other than being adaptive traits in these environments. We can't use the natural populations to answer this question, as there's tens of thousands of years of divergence between these populations, which creates large genetic blocks of inheritance. So instead, we use the recombinant population, which has small allelic combinations or haplotypes, to ask whether is there association between height and gravitropism in the genome. So we use the F11 control that was grown only in the glasshouse and therefore not exposed to any selection. Surprisingly, we found that there is no correlation between height and gravitropism. So this indicates that genes for these traits are not tightly linked in the genome and therefore are not controlled by all of the same loci. So next we asked whether natural selection could reconstruct the correlation after 3 rounds of selection in the dune and headland environments. So we found a weak phenotypic correlation between height and gravitropism after selection in the sand dune, and a strong correlation after 3 rounds of selection in the rocky headland, suggesting natural selection was likely very strong in that environment. So overall, these results suggest that natural selection creates a genetic correlation between these 2 traits and that their evolution does not rely on the exact same genes or genes that are very close in the genome. So far, these results suggest that local adaptation can arise from standing genetic variation that reconstitutes favorable genetic architectures. And given the physiological differences in auxin responses discussed before, this implies that genetic variation in hormone signaling can quickly diverge in nature and modify the architecture of plants to enhance survival. So now that we have found that gravitropism likely contributes to local adaptation, we began to explore the genetic basis of adaptation. In other words, what are the genes contributing to gravitropism in Senecio lautus? So using the F11 recombinant population where gravitropism was measured, we performed selective sequencing on the tail of the gravitropic distribution, where there is a 56-degree difference in gravitropism between the means of these two tails. So by comparing the genetic differences between the tails, we expect to find differences in genes underlying gravitropism. Reduced representation sequencing led to examining associations of 28,000 SNPs, or single nucleotide polymorphisms, between the tails of the gravitropic distribution. So the SNPs shown in red are the most differentiated between the tails. So these have an allele frequency change of more than 0.15, and they disproportionately contain homologous genes with predicted gene ontology categories of transport and localization of molecules within and between cells. So this is consistent with expectations, as redistribution of auxin is required for a gravitropic response. So I'll briefly talk about two of these SNPs above this line that lie within candidate genes that have predicted functions related to the auxin pathway. So these gene regions contain ENOD1 and ABA3, which have functions related to the auxin pathway as expected, but they also have functions related to salt resistance and reproduction, which could also be contributing to adaptation to contrasting gene and headland environments. So here we look at the effect of ENOD1 and ABA3 genotypes on gravitropism. Gravitropism angle is on the y-axis, and the two genes and their different genotypes are on the x-axis. So ENOD1 CC genotype, shown in blue, which is all individuals that have 2 copies of the cytosine allele, produces gravitropism more than the other genotypes. And ABA3 GG genotype also reduces gravitropism. And then when we look at these two genes, when they're co-inherited, they reduce gravitropism even further. In fact, ENOD1 and ABA3 explain 45% of the difference in gravitropism in the F11 population, which is a massive contribution to the phenotype. And this is in the same direction as the natural populations, where the headland parental population has high frequencies of both of these agravitropic alleles, and the alternate alleles are favored strongly in the June parental population. The results suggest that ENOD1 and ABA3 genes likely contribute to the evolution of gravitropism in these populations. So now that we've demonstrated that gravitropism has contributed to local adaptation, and the genetic basis of adaptation likely relies on divergence in the auxin pathway, we explored whether adaptive evolution of gravitropism is causing intrinsic reproductive isolation. So we asked the following question: if we crossed gravitropic individuals with agravitropic individuals, would they produce viable offspring? In simple words, no, not all the time. Almost a quarter of crosses between individuals with a different gravitropic response failed to produce seed. Relative to crossing success within each tail, this failure rate is almost 3 times higher than failure in the gravitropic tail, and while the agravitropic tail never failed. If we dive into the genetics of this to look at what alleles are associated with this sterility, we find that the ABA3A allele is positively associated with hybrid sterility, in which individuals with the gravitropic favored A allele had a higher percentage of failed crosses. This suggests that ABA3, or a tightly linked gene, might play a role in creating the observed genetic correlation between adaptive evolution of shoot gravitropism and hybrid sterility in Senecio lautus. So this shows that adaptive evolution can cause intrinsic reproductive isolation. So to summarize all these results in a nutshell, adaptation to 2 contrasting environments has led to adaptive evolution of gravitropism, and genes underlying this trait, or a very tightly linked trait, leads to hybrid failure. So these results provide a clear case where local adaptation can drive intrinsic reproductive isolation. So this research shows that evolution of hormone signaling pathways in plants provides a simple mechanism for the rapid adaptation to new habitats and to the incidental evolution of intrinsic reproductive isolation as a consequence. So some of the follow-up analyses we have done after this paper include investigating the relationship between gravitropism and salt tolerance in Senecio lautus. So we speculate there could be a physiological connection in relation to the effect of salt on cell turgor and how this influences cell expansion, which is required for gravitropism. So let me motivate this idea with some preliminary data. So firstly, in the natural populations of Senecio lautus, headland soil has a much higher salt concentration relative to the dune. So we found that sodium is 540 times greater in the headland soil relative to the dune soil, while chloride is 30 times greater in the headland. So we've also discovered that in controlled conditions of the glasshouse, headland plants have a higher survival after exposure to high salt. So these results that I'm trying to find the time to publish show that headlands have high salt tolerance relative to dune individuals. So we also know from research presented earlier that headlands are largely agravitropic and dunes are largely gravitropic. However, what we don't know is whether these phenotypic correlations we observe are also genetic. So to investigate whether these traits share some of their genetic architecture, Mariam and Zoe completed a small experiment to test salt tolerance on hybrid seedlings with divergent gravitropism values. So the experiment uses the recombinant population I talked about earlier, and we crossed hybrid individuals that were agravitropic together and those that were gravitropic together to produce F12 seeds. We predicted that the agravitropic parents would produce offspring that were more salt tolerant if genes for these two traits were tightly linked. Salt tolerance was tested in petri dishes where salt solution was pipetted into the petri dish every day for 29 days to test survival. So here we have the results from this pilot experiment, and the graph shows the survival curve where days alive is on the x-axis and survival is on the y-axis. So there was a significant difference in survival between the agravitropic seedlings compared to the gravitropic seedlings at the highest salt concentration. So this is consistent with our predictions. The agravitropic seedlings, the black line, had more individuals surviving than the gravitropic seedlings, indicating that the individuals that respond poorly to gravity stimuli are more salt tolerant. So gravitropism and salt tolerance share some of their genetic architecture. In other words, there is a genetic correlation between the two traits and therefore with fitness. So this correlation could either be via the use of the same genes, that is, they are pleiotropic, or different but very, very tightly linked genes. So what these preliminary results do not answer is if direct selection for one of these traits can drive the evolution of the other. So I'll address this question by conducting a salt tolerance directional selection experiment, where I can see whether direct selection for salt tolerance reduces the gravitropic response in the population. So ultimately, we hope to explore the molecular interactions that might exist among hormonal and stress pathways, so we can learn how networks module physiological responses when exposed to contrasting environments. So I'm currently co-advising 2 PhD students that will follow up this research. So Zoe is looking at gene expression differences underlying gravitropism and is hoping to explore connections between the auxin pathway and the ABA pathway. Abneet is building a genotype to phenotype map of gravitropism using the recombinant population I have mentioned before. So she'll look at whether this genetic architecture is repeated across natural populations and if they can be predictive of other phenotypes in populations found in similar conditions. And that brings me to the end of my talk. So I'd like to say a big thank you to my co-authors, and I couldn't have done this research without you. And a special thanks to my supervisor, Daniel, for guiding this long, long journey that finally led to this publication. Also, thank you everyone who contributed to the 3 years of field experiments and maintenance of thousands of plants over the years. And thank you all so much for listening. I look forward to your questions and future discussions on this topic.
Thanks, Melanie. It's very good. Okay. Yeah, so if anyone has a question, they can either do the raise a hand or they can type in the chat. I see people are clapping.
Thank you.
Well, while people are thinking, I might have missed this, but do we know the molecular reason why the ABA gene you had caused hybrid sterility?
No idea. I was kind of surprised to even find it at all. So ENOD1 actually has more associations to reproduction in terms of pollen tube reception, which is also linked to the auxin pathway. So I was thinking more along those lines, but in terms of ABA3, likely to do with salt or stress response. So really like to look into that further, basically.
Yeah, just off the top of my head, I can't think of a good reason why that would work, but I don't know that much about hybrid sterility.
Yeah.
Okay. Oh, there we go. Francis.
I was just curious, and I'm sorry if I missed this, if you already said it, but I think you mentioned that there were SNPs in ABA3, and I was just wondering if you've looked at what those are and how those might be affecting function of that gene.
So we're basing the functional analysis from Arabidopsis, and
Yeah.
Senecio isn't that well annotated, so I didn't want to draw too many conclusions basically from a SNP in the species so far distant from it. So I just looked at that this particular SNP that I was talking about just lay within the ABA3 and the ENOD1 within that gene region. So yeah, I didn't want to draw functional stuff.
No worries. Thanks for that.
I can see we have anyone else with questions. Oh yeah, you can go ahead.
Hi, nice talk. I was wondering, so how do you define fitness? How did you measure that? Maybe you explained it and I missed it.
No, I completely skipped that so I could cut down my talk. So fitness in the field with the transplant experiments is we combine multiple fitness measures. So first is, do they even germinate in that environment? And then the next was how many days can they survive? And that was, yeah, so they were the two key ones that we included into the model.
Oh, okay, cool. And you did the selection stuff, you did then in the wild, right? Somewhere, I don't know, on Strady or something?
Yeah, at Lennox Head and the Dune and Headland environments there. So they're quite close to each other. So I spent 3 months of my PhD at Lennox Head, so that's quite nice.
Cool. Yeah, thanks for that.